Okay, so this stress presentation will be about hydrolase. Hydrolase. It is an enzyme that breaks down bonds using water in a reaction called hydrolysis. Why is it significant? It's a fascinating group of proteins because they play a significant job in breaking down particles by adding water, making them essential for life. They assist with absorption, energy creation, and even helping in the separation, waste, in cells. Hydrolase has many great applications for the body, in addition to the fact that they can keep our bodies moving smoothly. But they're also utilized in medications, food creation, and environmental cleanup. For instance, some hydrolases assist with separating fats and proteins that I'll get into later, while others utilize in detergents or even separating plastic waste, which I also will talk about. The flexibility and significance in both biology and the industry make them an extraordinary protein to present on. This little graph right here represents all of what hydrolase is involved in the market. They have a global market size, as you can see, is $5.5 billion since 2021. It's at the bottom. It shows pharmaceuticals, cosmetics, as I said, detergents, textiles, things like that. So it's really involved in your everyday life without you even knowing it, especially inside when it breaks down, like I said, fatty acids or fatty materials, etc. So as far as its structure, the primary structure is the linear sequence shown on the right of amino acids linked by peptide bonds, creating a specific chain, determining the protein's unique properties like the overall shape. It serves as the linear foundation for folding in the higher structural levels like secondary and tertiary. The secondary structure, as you can see, there's a little bit of alpha helixes and beta folding, beta sheets. Shows clear beta sheet structures. The flattened arrows right here, as I was showing you, they're connected by loops and turns like alpha helixes. They're less prominent, but they're still present, like this one right here. Secondary structure stabilizes through hydrogen bonds between amino acid residues. The tertiary structures show the compact globular shape from the hydrolase protein. This arrangement allows the hydrolase to perform its function. The folding pattern shown is characterized by many hydrolyses, ensuring proper substrate binding and catalyst. So this whole entire thing right here with it has chain A also helps show and depict the tertiary structure. So there's many, as you can see, bonds that are made in hydrolase. We have hydrogen bonds, we have hydrophobic interactions, we have ionic interactions. Of course, we have the one that's always there, VDW, interactions with water or solvent molecules, and they ensure that hydrolase maintains its correct shape. And as far as the disulfide bonds, as you can see that are highlighted right here, it is a covalent bond formed between two cysteine amino acids within the protein structure that contribute in the proper folding and the stability of the enzyme itself. So as far as the function of hydrolase, they act like tiny biological scissors that help break down larger molecules by adding water. They play a critical role in many processes of the body, like I said before, such as digesting food, breaking down fats, proteins, and carbs into smaller pieces that we can use for energy. Some hydrolases also help remove waste from the cell and break down substances. As far as everyday use, like I said, laundry detergent is used in there to break down stains, or even in the industry, like I said, to help break down plastics. The main one that I wanted to focus on was lipase. It breaks down the fat molecules in glycerol and fatty acids. The water itself helps split the chemical bonding, holding the fat together. That's why I wanted to pick it right here. So you have the lipids, and then lipase is added, and it breaks down into glycerol and fatty acids. In the digestive system, lipase is produced in the pancreas. Then it goes down into the breaking down of the fatty acids. It catalyzes with hydrolysis by adding water, and then it's absorbed by the cell lining of the intestines and transferred through the bloodstream. So to connect the dots between function and structure, the function and structure are closely related with different protein domains playing specific roles. Hydrolase often has active sites where molecules can be broken down and chemical reactions occur. Some hydrolases also have binding domains, which help recognize the attachment to specific target molecules and stabilize those domains. They work together to ensure efficient breakdown of larger molecules. The one I wanted to focus on was, like I said, lipase, the fat digesting enzyme, has a pocket that fits fat molecules perfectly, allowing for the interaction between water and breaking down those bonds. Um, Cofactors stabilize those domains, helping ensure proper enzyme activity. At the bottom, I just wanted to depict how the hydrolase right here, hydrolase ion of breaks down protein into two smaller peptides right here. And then here are my sources.